All right. Hello, everybody. Welcome to the awkward period of Instagram Live where it's just one person sitting here talking to themselves. Hey, there's Dr. Pasca. All right. We are doing a live stream today with Dr. Sergiu Pasca. Hello, welcome, folks. If you saw the event or the video I posted last week about organoids and assembloids, we did a video about Dr. Pasca's work at Stanford University. He's doing just truly remarkable, cutting edge, impressive, a bit futuristic research. And so he's going to be joining me here to discuss that work. And I'm going to ask him a few questions to give you an opportunity to talk with him. Forgive me for staring at my screen here. I'm going to try to get Dr. Sergiu in here. Let's see. Thank you, Laura Leah, for your enthusiasm. I appreciate you being here. Let me see. Yeah, this whole Instagram, as a moderator, this whole Instagram live stream situation is complicated and always confuses me. Hello from France. Thank you for joining. While we're waiting for Instagram to work properly, where is everybody coming in from? I'm very curious. I know Instagram has a international reach. Hungary, very cool. Let's see, Dr. Pasca should be joining any moment now. I just accepted the request. California, anywhere near Stanford, Palo Alto, Sweden, Mexico? No way, this is really cool. Okay, Sergiu Pasca, unable to join. That's just perfect. Let's try again here. Utah, Germany, West Virginia. I did my undergrad in West Virginia, Killy, Detroit, Texas, Nigeria. Incredible. Okay, let's see here. Milwaukee. Contranta? I don't recognize that. Where is that? I've never been to Montana. Oh, wait. No, I've never been to Montana. I wish. I've heard it's gorgeous. Okay, here we go. Okay. You know what? Hold on a second. I might have to like reboot this or something because it's giving me a problem with Dr. Pasca getting in here. Let me see. There we go.
Finally, yes, it works.
Welcome, Dr. Pasca. How you doing?
Hi, good to see you.
Is that an electric?
Sorry, for some reason I was like being kicked out.
Yeah, no worries.
By the network.
Yeah. I'm glad we got it to work. Is that an electrophysiology rig behind you?
Well, maybe, yeah. Yeah, it is. I am in a room in the lab, hiding, I guess.
Well, thank you for joining us. I just want to start by just saying thank you, not just for being here, but for being open to this kind of thing, because the number of scientists who would be willing to do a live discussion open to the public like this on Instagram, on social media, is low. And I appreciate deeply that you are open to it. And not only are you open to it, but you're doing this at a really impressive level of your career. And, you know, for those joining us, I want you to understand that Dr. Pasca is pioneering some seriously high level research. And, you know, I'll go out and say it. I don't want to jinx it, but you're probably looking at a guy who's going to win a Nobel Prize at some point here. No. That's my opinion. That's just my opinion. But so really, thank you for being here. And
thank you.
Please, I guess if you want to start by introducing with all your long academic titles that you have.
Sure. Yeah. Yeah. Well, I don't know. I'm a physician by training, really, and to a large extent, most of the work that we've done has been geared towards trying to understand the biology of complex disorders of the brain. I'm a professor at Stanford. I'm a professor of psychiatry and behavioral sciences at Stanford, where I also lead the center that is to a large extent trying to bring people together with various technologies to, again, tackle this important problem of accessing the human brain in a non-invasive way that would allow us to understand the biology of these complex conditions.
And that's, what is that? The Center for Organogenesis? Is that what it's called?
Exactly. So, yeah. So that's the Stanford Brain Organogenesis program. Yeah.
Yeah, and for students, by the way, just be aware, Dr. Pasca, I understand that through that center you do training workshops and sort of, I forget, just describe sort of what that's like.
Yeah, I mean, when we developed some of these methods, now more than like 10 years ago, it very quickly became very challenging to train everybody who wanted, like everybody was willing to come to the lab and kind of like intern in the lab. And at one point it was just like impossible to accommodate that many people. So then we started this course, just kind of like a hands-on course. It's like completely free. People come from all over the world. We bring about 25 people at a time. And then my entire lab teaches that for a full week, and we pretty much show how things are done. Of course, the critical steps, these experiments are very long, so it's sort of like it's almost like a cooking show at the end because we kind of like have the parts already ready and we show the critical steps. But that has been incredibly enabling because now we've helped more than 300 labs around the world to implement these methods, and everybody's asking different questions, tackling human brain disorders from different angles. So that has been really enabling, I think.
That's amazing. So what you're essentially teaching is how to generate brain organoids. Is that correct?
Exactly. How to generate how to take the stem cells either from patients or stem cells that are from controls that are genetically modified, how to turn them into specific cell types in the brain, specific brain organoids that resemble this different regions. And now, especially in the last few years, with more and more interest in assembloids, how to put them together 2x2, 3x3, or more, and study some of the circuitry and apply them for disease.
Yeah. Well, let's do a little bit of that training here today. Then I'd love to have you explain, in lay terms, how you go about generating a brain organoid, and maybe actually start by sort of defining what a brain organoid is.
Sure.
And I think a key aspect to focus on is, where are these cells coming from? How do you end up with brain cells in a dish?
Yeah.
Human brain cells in a dish.
Yeah. So historically, the main challenge that we've had for us studying disorders of the human brain, both neurological and psychiatric, has been that the organ of interest, which is the brain, is really inaccessible. So unlike in any other branch of medicine, think about like dermatology or even think about cancer medicine, the tissue of interest is generally accessible. And as this revolution in molecular biology happened over the past half a century now, we've seen that most of the therapeutics that have been developed have actually come from accessing this tissue of interest and then applying the power of molecular biology to it. But of course, for the brain, we can just like open, the brain and like study the brain directly, and then of course we can get tissue after some of these patients have passed away, but then the tissue is obviously not firing electrical signals. Is much more difficult to study. To be honest, I believe that to a large extent, the reason why we've been so far behind in psychiatry and neurology versus any other branch of medicine is because, not just because the brain is very complex, and it is, but also because we haven't been able to access it. And so for the past 15 years or so, we've been trying to recreate in the lab in a non-invasive way some of these cell types from patients specifically, and then study them in order to find therapeutics. And that remains sort of like my main, sort of like my North Star, is trying to find therapeutics for some of these devastating disorders of development that come with seizures, with severe intellectual disability, and so on and so forth. And this was all this work was enabled almost 20 years ago when I was finishing my clinical training by a discovery made by a Japanese scientist who showed that you could actually take skin cells from any individual, and through some genetic tricks, you can actually push them back in time to resemble the stem cells from which like the entire organism develops. And so what we did is in those early days, I was still training. I came to Stanford and took some of this, took some skin cells from patients that have a very specific form of a neurodevelopmental disease called Timothy syndrome. And we took those skin cells, brought them to the lab, turned them into stem cells, and then developed in those early days, very simple, of course, at that time, methods to guide those cells to become neurons. In other words, we just like spike in the media, in the cell culture broth in which we grow them, growth factors, small molecules, and tell the cells to become neurons. And after a few months, some of them do become neurons, and they'll have electrical signals, and they'll look like a neuron, and they'll fire like a neuron. But then it also became clear that while we can find some differences in those cells, it's actually a lot of the properties of circuits are not actually captured in that preparation. Essentially, just think of it as at the bottom of a dish, kind of like flat, while the brain is three-dimensional, has a lot of brain regions that communicate with each other. And of course, we don't want to recreate the entire brain. It wouldn't be that useful, to be honest. We want a very simple preparation that allows to capture some of these early aspects of circuits. And so we started playing with aggregating some of the cells. So essentially just putting them in a bowl of cells. It turns out that if you do that simple trick, and now, a lot of labs have been doing that even outside of neuroscience, you can keep them as organizing, self-organizing. And today we call an organoid a three-dimensional cell culture system that is like an organ, so organoid, but it's not an organ. It's not the organ of interest. It's like an organ. They will have some of the cells that are present and maybe some of the functions of those cells. And so think of it as a bowl of human neurons and human astrocytes, so glial cells that resemble a specific brain region.
Yeah.
Um,
And
yeah, hopefully that makes sense.
Yeah. Oh, yeah. I mean, so since we've started communicating, not on Instagram Live, but just talking about your work, I've learned about the distinction, the important distinction between calling something a brain organoid and calling it a mini brain.
Yeah.
And a lot of people refer to brain organoids as mini brains, but it makes sense. They are actually not, in effect, mini brains, because there are, I mean, what is the actual scale we're talking about, and sort of level of network activity and diversity of cell types
Yeah.
in an organoid compared to a real brain, a human brain?
Yeah. So sadly, there's been, a misunderstanding or perhaps like an oversimplification that some of these cultures are brains in miniature. And they're not brains in miniature. They're not an entire brain in a miniaturized form. Again, they are visible. So, they're 3-4 millimeters in size. So they're relatively large for a cell culture. You can see them with the naked eye. But they don't have all of the cell diversity of the brain or all the circuitry. So, think about like, the outer layer of the brain, which is the cortex. It will have, it will resemble parts of that. Or it will be a little bit like a spinal cord. That's why we very often refer to them as regionalized organoids, meaning that they resemble regions of the nervous system.
Yeah.
And so now, we've shown that we can indeed make a lot of this, like, dozens of brain regions, but none of them are, perfect replicas of what happens. And that's, to be honest, like never really been the goal. Nobody wants to, create a perfect replica of that. We want to create a good enough model for those cells that are present in that brain region that would allow us to understand how disease develops.
And it makes sense because, when you're talking about someone who has, let's say, Timothy syndrome, a neurodevelopmental disorder, and, as a physician, you desperately want to understand what's going on in the brain, right? Especially if they have something like epilepsy, if they suffer from seizures, you want to understand what's going on with the electrical activity in the brain. But with today's techniques, the only way to do that is, as you mentioned, there's two ways. One, to do neuroimaging, put them in an fMRI or do EEG or something like that, where you're looking from the outside at very, not high resolution. You're looking at chunks of maybe millions of brain cells in each chunk of activity you're measuring.
Exactly.
Or if after they die, looking at the actual brain tissue, which is no longer active, which also has a lot of limitations. So what you are able to do in your lab, which is just incredible, and why it's so cutting edge and sounds kind of futuristic, is to take a skin swab of skin cells, turn those into neurons, grow them together into clusters of neurons that don't represent the entire brain, but represent a tiny little portion of the brain, and then study the actual live activity of those neurons, which are genetically identical, I would assume, to the neurons in that patient's specific brain. And, Then with assembloids, those who saw the video last week will understand assembloids is when you take multiple of those and combine them. And it gets especially interesting when you have what you've done, taking multiple organoids that represent different brain regions. Because, I'm curious, actually, I'll ask you right now, what happens if you take a cortex and a cortex? Do they kind of just cluster together versus what you've done,
Yep.
which is taking specific clusters representing different systems that would come together in the human nervous system, and it creates these whole systems. So I guess let me ask you that. What
Sure.
happens when you
Yeah.
take two cortex?
Well, I mean, in the beginning, to be honest, I mean, we know that the human brain or the brain in general functions through this complex interactions between its parts. So we knew from the beginning when we were building some of these organoids, 12, 13 years ago, that ultimately we will have to model some of those interactions. And initially, funny stories. We've spent actually quite a lot preparing for this. Like, how are we going to actually like fuse them or put them together? And I remember somebody in the lab worked actually for quite a while, trying to find all kind of biological glues and like stick them or like put some electrodes that will allow them until somebody else came and says, look, it's very simple. You just put them at the bottom of a tiny tube, you leave them there overnight, and next day they're essentially fused. Because what, of course, we forgot is that the cells are actually, to a large extent, coming with instructions on how to connect with each other and how to find each other. And so very often, even when we don't know the rules for how the cells are connecting with each other, they can actually find each other meaningfully. And that's in the first assembloids that we now published, many years ago, almost 7, 8 years ago. We've shown that if you put two parts of the brain region where one provides cells for the other, then the cells in one of these organoids, in the part of the assembloid that has the cells, will literally point towards the other brain region and start to migrate and jump towards it until they sort of like populate it. Or if there are like connections, the cells will project and find each other in a meaningful way. And so I think to some extent the fascination with assembloids has also been that they come with some instructions. Now, of course, they can't recapitulate everything. A lot of the processes in the brain are dependent on sensory input, on like input from many other brain regions and from the body. But there is a basic plan of these connections that we can actually put together. And if you put two cortical organoids together, by the way, it's not that trivial because you will need the corpus callosum, you will need the midline structures for them to know that that is the middle of the brain. So very often they'll project a little bit from each other, but they're not like that keen because it's pretty much the same. So it's quite interesting how we're deconstructed, and sometimes I'm not sure whether it's the best analogy, but you can think a little bit of like some of this assembly. It's almost like Lego pieces, right? Because we can put them together, except that they can only fit in a specific way. So you can't just like put them, if you put them in a different order, you just won't get a structure that you want. So it's actually quite important to think that there are some limitations intrinsic to the system.
Yeah. And to me, almost like a vehicle analogy kind of comes together where it's like, you got one part of the brain, which is like the engine, and then you get another part of the brain, which is like, the tire, the wheels, and another part of the brain, which is like the steering wheel, and it's like
Right.
you put those three things together, you get a semblance of a car, right? But if you
Right.
take the steering wheel and the brakes and the exhaust pipe and you put them together, like what do you have? You have a bunch of car parts sitting together.
Precisely.
Yeah.
Yeah,
And
exactly.
and I think a key thing to understand here, to share, to focus on, is when the news stories pop up about your research and people hear like, oh my gosh, they grew a corticospinothalamic system in a dish.
Yeah.
It
Yeah.
seems, the first impression might be that your whole goal is just to like do this wild thing and be like, look, we did it, but
Yeah.
that's not it at all, right? It's going back to that patient modeling example of being able to grow a part of the brain. Correct me if I'm wrong. The purpose of
Yeah.
assembloids is to be able to not just grow a single part of the brain representing that patient's brain tissue, but to study how specific systems work in tandem in
Yeah.
specific conditions, right?
Absolutely. And of course, in doing so, we've also started learning new things about biology of the human brain, right? Which in itself, hopefully, will be enabling for other studies. But yeah, I mean, my lab is always, I guess, is known primarily for building these tools. Paradoxically, I never wanted to be a tool developer. Actually, don't like building tools. I didn't get into this type of research because of that. It was just very difficult to study these disorders, these complex disorders of the brain, without getting actual access to cells, because we do need access to cells with very high resolution, molecular, cellular, to develop these therapeutics. And I think the therapeutic efforts for Timothy syndrome it's a prime example of that. That you've actually had to study this disease from all possible angle, and just one more time to reiterate, this is a very severe form of neurodevelopmental disease. It's associated with autism, it's associated with epilepsy, intellectual disability, but this patient are essentially unable to live a life of independence in any way. And very often they would die either because of a heart problem or because of some of the brain conditions, epilepsy, for instance. It's been just impossible to study. We've tried to study it also in mouse models, which is also another tool that people very often look at. But obviously, the human brain is very different than the mouse brain. And so, while we've learned a lot from animal models, it is also clear that there are some aspects of human brain development and function that we won't be able to see in animal models. And I think that's where we're coming from. But of course, in doing this research, and I think you pointed out, there's a lot of miscommunication, sadly. Some of it gets amplified on social media. I think the example with mini brain is, which I've been, trying to fight as much as I can, convincing others not to use this term, because it's not just inaccurate, but it's actually, in my opinion, is even demeaning to some of the patients who are participating in some of the studies. And it's just simply not scientifically true. It's not a small brain in miniature. It's not. But of course, there's a lot of discussions now also what it means to actually build some of the circuits. And I know there's been some questions too about this. There are all kinds of ethical issues that surround this type of research, especially as we move forward and these models are becoming more and more complex.
Yeah, and by the way, just to the folks here, please feel free to drop questions in the chat as we're talking. But one of the questions, well, first let me just say I appreciate what you've said about the misinformation, and it's challenging because you're leading this research and then you're not in full control necessarily of the media
Yeah.
coverage and the way people talk about it online. And so again, I appreciate that you're interested in having these types of discussions because you can confront it directly. And it's funny because it may seem sort of inconsequential to say, hey, we shouldn't use the term mini brain. It's like, well, who cares? Why not? But if you think more deeply about it, you realize, yeah, actually, if you refer to it as a mini brain, that gives the impression that this is maybe actually representative of a brain. And then people can start to wonder, well, is it conscious?
Right.
Is it thinking? Does it feel? And then it brings the ethics really into the discussion, where I know that is a very important focus of your work, and you do a lot of work to bring more discussions about the ethics and make sure that the research is done ethically. And that is a small thing that may seem inconsequential, but actually really does change the public perception of the research. So let's talk a little bit about the ethics, though. I mean, one of the questions, someone actually dropped it in the chat. When you have an assembloid, you are moving more closely to a complete representation of a brain. It's no longer a cluster of cortical neurons. Now maybe it's a system. At what level do you think about consciousness entering this type of system? And
Yeah.
I mean, just to also clarify, you don't believe assembloids are conscious, right?
Yeah, I mean, and there's been a lot of discussions about whether there's any form of sentience or consciousness. Let's say sentience. Let's use the term sentience as sort of like a subjective experience. It's probably, I mean, I'm sure you know, and probably many in the audience know that even defining consciousness or sentience is actually not a trivial matter.
Yeah.
But generally, it is agreed that it's some form of subjective experience. And we don't think that we have, not even close in any of the circuits that we've built so far, some of the complexity of the circuits that are thought to be important for sentience. Now, I mean, there's just so you know, there's no, while we may have built some sort of sensory input in some of them, or maybe we've built some sort of outputs, it's never really been built in in complete, right, like both sensory processing and outputs. There are a lot of things that are missing. There's now blood flow. Many of the brain regions are sort of like missing. So I think there are a lot of components in this preparation that are still far away. Now, of course, when you transplant this into animals, and some, we and many others also have also done transplantation experiments into animals, which are sometimes necessary because we know that in a dish there's a limit to how much this cells can actually mature. So some of them really get stuck almost like in development. So that in itself is also another reason to believe that some very complex processes of cognitive processes are unlikely to arise in that preparation. Now, of course, we don't know, and we can never really say that they won't. So we always want to keep an open mind, and that's why we have a lot of discussions about this. But maybe some of the discussions are a little bit more relevant for when we put some of this into animals, and I think that's another. But we started, I've been preoccupied by these issues from the beginning. Of course, all the experiments that we do undergo review. So, we don't do them sort of like on our own. There are committees here at Stanford that are like looking at this experiment. In fact, we now have like even more committees and more discussions. We have an entire meeting. We've had a meeting at Asilomar, the historic place where some of these ethical discussions have started like last year, and we're going to have another one this year where we like really bring not just like us, the scientists who are doing the work, but actually like patient advocates who are thinking about the need for what needs to be done, philosophers, sociologists of religion, legal scholars about the legal implications of this work, and we've had some, incredible discussions about both so like the promise and what could, may not go well with some of these experiments. And that's why this community coming together, it's so important for having these discussions. And I think it's important to convey to everybody these experiments are not happening in complete isolation. They take years. Everything that you see published now probably started 5-6 years before, if not longer than that. And there are constant discussions about this type of work.
Yeah, and that's amazing that you're doing all of that work. And obviously, it's been very obvious to me in our offline discussions that this matters to you. Maintaining open discussion around ethics, and to sort of what I've seen in our conversations is that you are the person who pioneered assembloids. You are the first person to make that work, and now that it's spreading not just in neuroscience but to other fields as well.
Yeah.
And what I really appreciate is, you're both doing work to enable other labs to follow suit. Like we discussed at the beginning, your center at Stanford is teaching free courses for other scientists to come in and learn. But at the same time, you're also recognizing that there are ethical considerations here, and you're sort of pioneering this parallel effort to make sure that discussions are happening around the ethics involved, because it is such a unique and it is a very ethical, there's a lot of ethics involved inherently in the work.
Yeah.
So
Yeah.
it means a lot to me to see that in you, and I appreciate you doing all
Yeah. And as you know, I mean, all work, I mean, that's the paradox of it. If you think about it, like we were realizing more and more that brain disorders have uniquely human features. And of course, we also know that the models that we use to study them need to be more human. And obviously, the more human they become, right, the more uncomfortable we feel because they're closer. So I think that's why we have to have these discussions really, together as a community. And, really the public is involved, is going to be in the next meeting, is going to be involved. Journalists are going to be involved in this discussion. So this is not happening in complete isolation. I'm not a fan of that. But I think like, a lot of discussions will be useful. Something useful will come from these discussions. And in general, like, and you've been at Stanford, you've trained here, that the spirit here has always been to share. Everything that we build, we just share openly and free and as soon as possible for others to do. And again, I thought that, there's so many conditions of the brain that need to be tackled. I mean, hopefully my lab will work on a half dozen. And if we're lucky, maybe we're going to make a difference for some of these patients at one point. But there's so many other conditions. So we thought that the only way to do this will be for as many people to learn this technique and to implement them. And you'll be surprised, like, some people apply it to ALS, and some other people apply it to viral infections of the nervous system, and applications that we couldn't even like imagine, to be honest. And that has been, really, really powerful.
Yeah. Now, and for those who aren't aware, I just want to kind of plug your work a little bit more, Dr. Pasca, that, you mentioned, what, 10 years ago that you created the first brain organoid with Timothy syndrome cells. Is that right? Was it 10, 15 years ago?
Yeah, so we've done some of those experiments. So I think we've done experiments with Timothy syndrome neurons almost 16 years ago. So soon after Yamanaka made the discovery, we made neurons. They were in the beginning at the bottom of a dish, and then about beginning of um, uh, 2013 or so. Actually, we published some of the first examples, and the cells were already in 3D. At that time, the term organoid did not exist. It was not really used. That also was a term that came later, which, by the way, is probably not one of the most fortunate names. I wasn't necessarily a big supporter of like the use of a term that kind of like suggests that it's organ-like. But I think it's okay as long as we all have a clear definition of what that is. That is not a full organ.
Yeah.
But yes, I mean, it's been a long journey for Timothy syndrome, to be honest. It's been taking us now almost 16 years of studying this disease, and it took 12 to 13 until finally we understood enough of the biology that we could see where we could bring this therapeutically. And that's what a fraction of my lab now is primarily like focusing on, trying to translate this work. And my hope is that we will run a clinical trial in the next 6 to 12 months.
Yeah, and that's incredible. And yeah, for those who aren't aware, I mean, what you've done is not only modeled these brain systems in vitro in a dish, but you've also been able to study the biology, pinpoints the specific mechanisms that would have to be targeted to reverse the alterations you've seen, developed a potential therapeutic, and tested it and validated it in human brain cells. And now you are, like you said, advancing to the stage of potentially doing clinical trials. And so I think it's important for the general public to understand, and for those here to be aware that, as you're pioneering this incredible research, you're also developing new potential legitimate therapies for rare genetic conditions. And that is something that would never be possible if all we had were fMRIs or something like that. It was necessary to get in and model and study the biology. And that's just, it's unbelievable. And it's exciting because it's the first condition that you've done this with. And now, as this tool that you've created is becoming more widely adopted, it's opening new avenues for this type of study to spread to other conditions, like you mentioned.
Yeah, absolutely. And even since last year when we published this paper, the last paper on Timothy syndrome, which was the most kind of like translational of all, I mean, even since then, we've helped so many labs already kind of like implement for their own disorders that, because essentially the model there was like, we do as much as we can in a dish with patient cells, like actual patients that we plan to treat, but without actually involving those patients' brains in any way. Right, doing all of those experiments there, including safety study. We can do a lot of safety study in vitro. We can show whether the neurons are reacting in any way to that specific drug. Of course, we can exclude everything, but then you can even put them, and that's what we did in this case for the first time. We essentially put some of this patient cells into the rat, the neuron, and then treated the rat the way we would treat the patient, but look at the effect on human cells in an in vivo environment, so an actual organism, which is sort of like a necessary step before you move safely into patients. And so, I mean, I think this kind of like illustrates that we now have a platform where we can take, all the mechanistic steps all the way to, and in a way, it's personalized medicine, right? Because these are actually patient cells. And I think there was a question I saw a few minutes ago: are these genetic models? Yes, they are. They're exactly the patient cells. They can be exactly the patient cell that you want to treat. You can study in addition, develop that therapeutic specifically for that individual. So I think that's also one of the advantages of this approach.
What's unbelievable to think about is, it's steps away. You're at the you're doing basic translational science in a research lab right now, but in practice, it's amazing to think someone with a neurological condition could go to the doctor, and instead of them saying, hey, we got a couple of drugs that might work, we might, you want to try them, there might be side effects, whatever, yada yada. That if I'm the patient, my cells could be swabbed off my skin. You could grow a brain organoid in a dish, apply the drug, and test whether it makes a difference. While I'm at home making dinner in my kitchen and relaxing and
Yeah.
not having any effect of the drug, and getting a preview of whether or not it might target the mechanism and reverse whatever changes are happening in the neurons. That's unbelievable to think about.
Yeah.
And whether that's possible at a at scale with patients, it would be complicated. There would have to be a lot more labs like yours out there doing this type of work. But I mean, it is incredible, and it's, like you said, precision medicine.
Yeah, and we're getting closer to that. And this type of work can also be done for cardiac conditions, right? For heart problems. So you can actually make a tiny heart tissue from an individual that starts to beat in a dish, literally, and test drugs. Let's say there are like drugs that may have unanticipated side effects, or there are, let's say, cancer drugs that have side effects on the heart. Some patients react really badly to that. You can test that a priori. So I think just in terms of safety and having personalized treatment, I think this is certainly, now it's a long process. It's a long process, and if anything, what we've discovered is that even the timing of making some of these cultures recapitulates the timing of making them in actual real time in utero. So I think that one of the challenge will be, like, moving forward, how to accelerate, how to condense the process to a shorter period of time. But many labs now working on this. Somebody will figure it out. So we'll see.
Yeah, probably a more realistic model is, instead of like, hey, I have a specific condition, let's create an organoid just for me and test it. More like, let's start with genetic testing,
Right.
and there will be more and more research understanding how specific genetic risk factors change the function of the brain. And that research will be done in organoids and assembloids,
Yes.
which will inform the treatment
Yeah.
options. And then you don't have to make a whole organoid for me. You could just look at what my genes suggest that might be helpful for
Yeah.
me.
And one of the questions will be, how early do we actually have to intervene for some of these conditions, right? Or in other words, like, how early is too late? Because for some of these conditions, there is irreparable damage that is being done to the nervous system. We could say we don't know yet. I mean, the human brain is remarkably plastic during development. And so one of the key questions that we're also trying to answer is, how early do we have to deliver this therapeutics in order to reverse as much of the function of the brain in these
Mm-hmm.
patients? And I think that is still an open question. Nobody really knows, to
Yeah.
be honest, at this point, for neurodevelopmental conditions.
Right.
And then, of course, there is for neurodegenerative conditions as well, because one of the potential for some of this transplantation is to replace some of the cells that have died. Think about, like, Parkinson's disease. For Parkinson's disease, there is a clinical trial that is now ongoing. The first phase has been completed, where the neurons, the dopaminergic neurons that die in this patient, have been made separately from the stem cells, not from that individual so far, but from somebody else, and they're being injected directly into the striatum, inside the brain, and it seems to have some positive effects so far. So the potential for the cells is also to replace some of the cells in the brain that are missing. So really a lot of work to be done, and I really hope that, I'm sure there are many scientists here, or many who want to become scientists. I really think there's never really been a better time to do neuroscience than now, to be honest. I mean, the possibilities of studying the human brain, for understanding these conditions that are devastating for patients, the impact that you could make in joining this field. So I hope many of you will consider becoming neuroscientists, because this is really the best of times.
Absolutely. I hope so too. And I know there have been several messages that have come through saying, basically, Dr. Pasca, I'd love to work with you, keep an eye out for my PhD application, things like that.
Sure.
But, I want to ask you a few questions. And again, if you're in the chat, please drop some questions. One of the questions that came in on the video in a comment that I've been curious about, I think it's a really great question, is, you're talking about developing these organoids from specific genetic lines.
Yeah.
What happens, or have you tested, if you take two organoids from different genetic lineages? Do they form assembloids just as easily?
Yeah, that's a very good question. So we call that a mosaic assembloid, or a mosaic organoid, because it's sort of like a mosaic, a combination. And as maybe some in the audience know, we very often are ourselves mosaic. So very often we have cells that have mutated very early in development. There's also some evidence that babies and even us get some of the cells from the mom early in development. So we all carry other cells or other cells that are not genetically. So we know that cells that are not genetically identical can cohabitate, especially when there's not an immune response specifically as it is for us in a dish. So you can absolutely mix them. Generally, the rules for assembly of the circuits surpass the rules for like inter-individual, kind of like the inter-individual differences that are relatively minor in respect. But the reason why we use this quite often is because let's say you have two brain regions, and one is projecting into one side, and the other one is receiving the input. And the connectivity, it's unclear, it's not forming well, let's say in one condition. And we want to understand, is it because of this brain region not projecting right, or is it because of this brain region not receiving it? So one thing that you can do is now you can build an assembloid where either half is, control and the other one is patient. And that we've used that actually quite well in the past, and others have done that too. And you can disentangle where the defect is coming from, and that helps us again to point out closer and closer and closer to the molecular mechanism for that disease. So it's really another arsenal, tool in the arsenal that we have now for tackling these diseases. But yeah, hopefully that answered the
Yeah,
question.
that's brilliant, and I hadn't even thought about that, but it makes sense. It's like, you're trying to study necessity and sufficiency, you're trying to do
Yes.
science, and so you sometimes it's useful to say, okay, well, let's take this out of the question. Let's use a, non-genetically altered organoid for part of the assembloid and see if the
Yeah.
problem persists.
Yeah.
That's interesting. Another common question that I've seen, and again, folks, if you have questions, now's the time to ask them. Is about Alzheimer's disease. And,
Yeah.
people asking, could you use this to study Alzheimer's? As a person who has never done any organoid research myself, I would imagine that organoids don't live long enough to suffer from
Right.
Alzheimer's, right? Is that
Right.
true?
Yeah, so that's true. And we've shown over the years that actually the timing of development is actually quite well conserved, and cells know where they are in time. So, and there must be a reason why Alzheimer's disease, even if the biological onset of the disease is like 20 years before the clinical onset, it will still be for somebody in their 40s or 50s. So that poses some challenges. On the other hand, there are ways of like thinking, and a lot of people are trying to approach that to try to accelerate the progression of the disease, and sometimes that is done through studying genetically defined conditions. So maybe some know this and maybe some don't know this, but for instance, Down syndrome, the neurodevelopmental disorder of Down syndrome, where you have an extra chromosome 21, they actually develop early onset Alzheimer's quite often, because they essentially have the gene that produces the amyloid that is being deposited in Alzheimer's is actually on that chromosome. So instead of having two copies, they have a third copy. So now they have even more of that. So not all patients, but many of them develop that. So as you can see in this case, we can use this trick, and people have done experiments and have shown indeed that Down syndrome cells do produce more of this amyloid. And so that's one way of tackling that disease. Another way is to stress the cells. So sometimes you can expose them to very strong stresses in vitro, of course, in a dish, oxidative stress, radiation, all kind of other tricks to really see whether we can accelerate the development of that disease. So I think there's a lot of potential in doing this down the road. But of course, being late onset disorders and this cultures being early in development, it's a little bit more challenging. But that's a great project for somebody to do, honestly, and tackle this, scientifically in a PhD and accelerate the maturation of the cells to match that of a 50-year-old brain.
Yeah, that's fascinating as well. So I want to go back up. Someone asked a question. This is a tough one. Considering the current modeling capacity of brain organoids, do you think these systems could evolve in the coming years to mimic cognitive functions at a more complex level?
Yeah.
Difficult question.
Yeah, I think it will be difficult. It's difficult. Also, probably we have to define that a little bit better. Like, what would be a satisfying? It's hard to even know what would be a satisfying answer. What would be a cognitive process? Of course, these neurons in our preparation and especially in assembloids, they do fire action potentials. They do connect with each other. They have spontaneous activity. So you could even argue that some sort of like computation is taking place. We don't have that much evidence for that, but there are some signals that are being transmitted. And so I think the first question will be what level of computation is really possible with the preparation of the sorts, and especially when you have an input and you have an output, right? So you have some sort of like a readout that would be important. It's much more likely that an organoid will contribute to a cognitive function within the context of an organism where there is a behavior. There can be a behavioral manifestation because obviously these organoids also don't have necessarily complex readouts,
Yeah.
right? And so I think it's worth keeping in mind that there are limitations to what can be done with these cultures. But in terms of computation, I think that remains to be seen how complex it can be.
Yeah, it's interesting because it's almost, there are so many scenarios where studying brain organoids is advantageous relative to using a mouse model, right? Because with brain organoids, with human brain organoids, you're having actual human brain cells, since you can study
Right.
the molecular biology in a way that's much more directly related to what's happening in here. But with
Right.
something like studying cognition, it may actually be favorable in certain instances to actually just study mouse models because you have a living,
Absolutely. Yeah.
active
Absolutely.
organism
Yeah.
that you can put. Sorry, my dog has entered my room and grabbed my slipper and is now ripping it apart at my feet.
Yeah.
So yeah, so it may be beneficial in certain instances, and that's kind of a good point to consider when looking at that question of can it handle cognitive functions. I keep seeing there's this whole Pong study,
Oh,
and I'm sure you know what
yeah.
I'm talking about, where they put brain cells on an electrode grid and then they basically train them to play Pong, and it gives this impression that the cells are sentient and that they are watching the ball and going, darn, I let it go past,
Sure.
I'm going to play harder. But they're not, right? They're responding to inputs, and I think
Right.
that kind of thing has been misrepresented a lot to suggest that
Yeah.
these cells are active and thinking, but they're not. They're simply responding to specific inputs.
Absolutely. And I think, we tend to so like attribute, some of us, including scientists, I think, try to attribute like ever more complex features to some of this culture. So, just think about it. Like, just to give you an example, let's say, you can make a retinal organoid. People have made a retinal organoid, meaning that it has some of the retinal cells, including cells that respond to light. And let's say you put light on top of the retina and you see activity. You will say that the retina is reacting, but you wouldn't say that that retina is seeing anything, right? But it's easy to kind of like fall prey to that, logic and say, oh, the retina is like now seeing. But we know that seeing involves much more processing, right? With the thalamus and with the cortex, and it's a perception, and it involves many other brain regions in the cortex. So I think the same thing, and if anything, with cognitive processes, that it's even more complex, right? I mean, it could involve the hippocampus and various parts of the cortex, and none of those are really yet present in any of these preparations. And as you pointed out, animal models are very often much more useful for studying that process than this. And this work that we do with like this human stem cell models is here to kind of complement some of the work that is being done with animal models. So it's not here to replace it or in any way.
Right. Yeah, I think it works very well together. And sorry for if I look distracted. My dog is now actively ripping apart my favorite pair of slippers about 4 feet away. So I'm trying to get her under control while also managing this discussion. Um, actually, this question just came through because it's very sort of personal and scientific. How to convince my PI that brain organoids are the best way to test new therapies in, well, very specific monogenic rare neurodegenerative diseases. What do you have a response to that? Sorry, and please ignore if my dog
What
is
was the
racing all over the
first
place.
part? I missed the first part of the question. What was the?
How do I convince my PI that brain organoids are the best way to test new
Oh,
therapies
oh,
for?
oh, convince your PI. I'm not sure whether it's the best. I'm not sure. Whether that is the case. Always. I mean, I think we've understood a lot of conditions without the use of brain organoids. Yeah, I don't know what the best approach would be. Maybe convince the PI to comment one of these conferences that we have, at Cold Spring Harbor and other places, and really see how exciting some of the work is.
Yeah.
Or go ahead and get yourself trained in some of these techniques and do it during your postdoc. I mean, and these methods are here to stay for longer, and I think acquiring some of the skills if you're a scientist is probably going to be a useful thing.
Yeah,
Yeah.
absolutely. So do you have a next event coming up through the Organogenesis Center?
Yeah, we do. So we've gotten like an amazing like new cohort that is coming in early May, and then we're gonna announce a new one like sometimes in the fall. A competition. We usually give a lot of time because, yeah, I mean, people need to apply, and then we have a committee that looks at the application. We have hundreds of applications now, and then one of the things that happens is that some of the students are coming from far away. They need a visa. They need to secure like that visa, be able to come here and like train. But usually like the course takes place in the spring. The applications start in the fall for those who want to apply, and then the conferences for the field are usually in December every year. So this December we will have the first conference on assembloids at Cold Spring Harbor, and that would be across. And I think you pointed out this at one point that initially the term assembloid, when we introduced it, it was referring to the brain, combining different brain regions. And then quite interestingly, a lot of other people were like not studying the brain, started building assembloids for other organs. There are like bladder assembloids now, or they're like kidney assembloids. And generally it refers to three-dimensional cell culture systems where you have cells that are generally born in different parts of the organ or the body, and then they come together somehow. And so I think that's what we generally refer today as assembloids, as like three-dimensional self-organizing cultures that have a large cell diversity and different lineages of different origins. And so this December we'll have the first meeting on assembloids at Cold Spring Harbor, where really I think the brain is only a third of the meeting, if any. And then we have one on human brain development and 3D neural systems, so organoids, assembloids, and transplantation. That just like happened in December. We're gonna have another one in 2026 in December.
Excellent. Yeah, well, I know that you're always doing lots of lectures and
Yes,
workshops
of course.
and
Yeah.
sorts of things. Because someone also said, do you maybe plan workshops or lectures in Europe? I know that.
Oh,
And I,
yeah. Like I mean, workshops less so. I think it's much more difficult. We considered at one point like having some of this courses somewhere else. It's been very difficult. I mean, the course is also, by the way, taught by people in my lab. I mean, literally, like my entire lab, just does this for about a couple of weeks a year. They start their own experiments and they just like share their knowledge with others. It would be very difficult to do that somewhere else. And plus, of course, we have the cultures that we have to transport. So we haven't been able to do it somewhere else. But of course, I lecture quite often. Actually, in a couple of days, I'll be in Edinburgh and give a talk there for those of you who are in the UK. And then coming to Europe quite often and to Asia as well. So hopefully we'll meet many of you who are scientists like at various conferences.
yeah, excellent. Well, Dr. Pasca, I'm going to ask you the most difficult question that I always get asked in podcasts before we close out. Is there anything that you wanted to discuss that we didn't have a chance to discuss?
I think we covered the quite broad array of like issues, right? Like both kind of like and hopefully it became clear for more, because I think that was like maybe a little bit confusing for some, like what the motivation for this work is.
Yeah.
Which hopefully is more clear now that it is about understanding these conditions, and we're talking about devastating conditions of the brain, really severe neurodevelopmental conditions, not not mild conditions of the nervous system. We're not talking of variability in neurobehavior. We're talking about really severe conditions of the brain, and hopefully many got an understanding of how exciting this time is for tackling this with some of these models in combination with many other models that exist out there.
Well, I appreciate not only the great research that you're leading, but also just you for joining me here and, participating in this discussion and allowing folks to come in and ask questions. So keep up the amazing work. I hope I get to bump into you at one of your amazing lectures. I got to see you at SFN.
Oh, yeah. Yes. It's great to see you. Yeah. And thank you so much for what you do for talking about science and neuroscience in particular and sharing. I know it's not easy, what you do, but now that I get to interact a little bit more, I see just like how difficult it is for many of us, really to put the work that we do in more broadly accessible terms and really engage the public. I think the public should be engaged, should be part of this research. Science is it's fascinating. Everybody should be part of it, at least as part of discussions.
Yeah, right on. And someone said this work is important. I hope you host more video chats like this. I absolutely, I would love to. I think it's great to get researchers out of their laboratories and get them here on the internet to talk about it. So yeah, no, I really appreciate it, and thank you to everyone who joined and asked questions and had great comments. And yeah, Dr. Pasca, thanks so much again, and
Thank
maybe we'll
you.
do it again sometime.
Absolutely. Thank you so much. Thank you. Thank you.
All right, take care.
Thank you.